Hey class, welcome to Chem 714 Pharmaceutical Analysis. I'm your host, Professor DePaolo, and this will be the first of several videos that are going to be overview videos to get you kind of assimilated with each unit before we go over it in class. And they'll cover really the basics of each unit, and I feel like it'll be a good study tool when it comes time for exams. So in the first unit of Introduction to Pharmaceutical Analysis, we're first going to be going over the different properties and principles that are governing the pharmaceutical analysis process. So we want to be covering the different methodologies and also what are we actually doing in pharmaceutical analysis. So the purpose is to identify substances, purify them, separate them, quantify them, determine their structure, and also the concentration in these pharmaceutical analytes. And this is very important when it comes to the efficacy of drugs and the manufacturing of drugs when we get into the clinic or even from the earliest drug discovery phases. The purity, the structure, making sure the integrity of the substances is intact and it's almost 100% pure. This is very important. And we like to keep an eye on and have analytical methods to determine the chemical structure at all stages, whether it be from preclinical in the cell or even in solution in vitro work, all the way to the mouse, to the clinical trials, to manufacturing in large batches. We want to make sure that the chemical structure is intact and that there are no derivatives or different impurities that are in the sample. We want to also keep an eye on the amount, make sure the concentration per dose, per batch is consistent with the protocols that we have in place. And of course, along the way, we want to make sure the mechanism of action is as the drug does what it's supposed to do. There are certain workflows in place to extract certain contaminants using different solvents that they dissolve in, but we'll go over that in more detail. For those of you in this class, you are in a great spot to be in New Jersey because it is the largest biopharma hub in the country. We have the biggest companies. Most of them are headquartered here in New Jersey. And even if they're not headquartered in New Jersey, they have huge labs with a lot of employment opportunity. So the course objectives, we want to go over different instrumental techniques and also go over the regulatory processes involved in the pharmaceutical industry. So the major kind of umbrella techniques we're going to cover. We have chromatography, which is there's different separation techniques involved in chromatography. Usually in pharmaceuticals, they are paired up with mass spec. So we want to separate our analyte, determine what is in the mixture, and then directly quantify it based on its mass. That's where we use usually an LC-MS, for example, or GC-MS. So gas chromatography, mass spec. So we use these techniques in tandem a lot of the times. So in our screening workflow, in our QC workflow, we're going to be using pretty much all of these in most pharmaceutical projects. We have different spectroscopy techniques, which are either qualitative or quantitative. We'll talk about that in a minute, to determine the structure and the functional groups within the structure. So we have UV and UV vis. UV is usually UV vis, so 200 to 800 nanometers in length, and we're using light to interrogate matter. So that's basically the principle. And this will be covered in Unit 2. We'll also have Raman, we have fluorescence, we have atomic absorption, and we have NMR, which is more radio waves and magnetic frequencies. So then we have spectrophotometry. So spectrophotometry, which is more of a quantitative method and that is used to determine concentration. We have different biophysical techniques, which are used to interrogate the mechanism of action and whether the compound or analyte of choice, the drug, the lead drug, binds to its intended protein or antibody target, whatever the target may be. We have different techniques for this as well. We have biochemical techniques to do the same, but not only to detect if the analyte binds to its target, but to make sure that target goes through the conformational changes that it's supposed to create a cellular outcome. And also, as mentioned, we have mass spec. So we want to explain the application of these techniques in this class. We want to cover the regulations associated with the pharmaceutical industry, that being GMP for the most part, so good manufacturing practices, and also basic and good laboratory practices. So we have a few of these. So we have GMP, We have cGMP, which are, they're used interchangeably. We have GLP, which some of you might be familiar with, as there are many GLP labs in New Jersey. We have GCP. So if you're involved in clinical stage projects, you have good clinical practices that you need to follow. And we have good documentation practices, GDP, not guanine diphosphate, which I'm used to seeing GDP as guanine diphosphate, but it is good documentation practices. So that's standard SOPs and so forth. And we have GXP, which is just, good practices in general. So we're going to cover the different aspects of GMP, that being the products, people, processes, procedures, and premises. So that's not three P's. That is the 5 P's, and we'll cover that more in detail. We have our standard operating procedures, our SOPs. So this covers really anything involved in an assay or any analytical technique, from the sample prep, the pipettes that are used, the serial numbers of the instruments used, the different settings on the instruments, the analysis protocol. So after you generate the data, what are you doing with it? This is what the SOP has, all of this. So here's an example of an SOP that I created at my former company that goes over one of the techniques that we'll cover in this class, called dynamic light scattering. And it's a way of really interrogating a sample to determine its purity, its aggregation, its precipitation, how it behaves in a solvent, and also the concentration. Well, you can kind of determine concentration. It's really a qualitative technique. So you have everything from preparing the buffers, the dilutions of the compounds, the controls, the blanks, the instrumentation, the consumables. Really everything is covered in an SOP. For a really good SOP, it's important that it's very thorough, and the point of it is to make sure someone else can follow it perfectly. So I am no longer at this company. Someone has this SOP, and it's their job to figure out what I'm trying to do and what I did, and to reproduce my work. That's the idea of an SOP, reproducibility. So GMP and cGMP, they're really the same thing. So the 5 Ps, we talked about that. Quality assurance versus quality control. Quality assurance is the umbrella term incorporating quality control. We'll talk about that in detail. We'll talk about the FDA a little bit. So the FDA presides over the release and the approval of any drugs used in the clinic, and also food and other certain cosmetics. Not every cosmetic, but certain cosmetics. Then there is qualitative analysis, which is to identify the constituents or the functional groups or structure or what is in the sample, not necessarily how much of it. That's what a quantitative analysis is. So the measurement of the quantities of the constituents present in a substance. And there's different techniques for both. Some techniques do both, but we'll learn about those techniques. We also want to focus on accuracy versus precision, sensitivity of an instrument, false negatives, and all of that. So we'll cover all of that in class. We want to cover also selectivity, accuracy, and precision. A lot of these might be review for you, but we'll cover that in class. So the importance of different blanks and controls in any experiment. We want to have controls, a positive control, a negative control, different reagent blanks, method blanks, and we'll talk about those. Accuracy versus precision. So accuracy is basically how close the value that you're obtaining is to the real value, the true value that you're expecting. So if you want to calibrate your instrument and you have BSA, which is a standard bovine serum albumin, and you want to use this assay to detect the concentration, which is or the mass of it, which is commonly done. And the accuracy is how close that number is to the real mass of BSA, which is around 66 kilodaltons. The precision is how close your measurements are to each other. So if you consistently get 61 kilodaltons and the true value is 66, your precision is good, but your accuracy is not. So this can help you determine a systematic error versus a random error. So in this case, that case that I just said would be a systematic error. And you can fix that with a setting probably. So we'll cover that. Dynamic and linear ranges. Calibration curves are important in quality control and in using your controls. And to identify and to be precise and accurate in your measurements. We want to define outliers as well. We will talk about signal to noise ratio, and the golden rule is your signal to noise ratio should be greater than 10. Some assays accept the value at 5 or more, but I say always greater than 10. Here in our example, it's 16, which is good. We're also going to cover the boring stuff, that being significant figures. So it is very important. But, once you get into the pharmaceutical industry, you don't have to remember, quote for quote, your sig fig rules. You will implement them into your assays naturally. You're not going to record 15 decimal points. And that's just something you'll do. Usually it's 2 or 3, depending on the instrument. So we'll talk about sig figs. That's a lot of it. Rounding. And that's really basically it for the first lecture. So we'll cover different types of error as well. And also, we'll talk about the different phases of clinical trials. Why is this important? And the reason why a lot of these methodologies and these governing bodies of our SOPs, of our practices, and our documentation, the reason why they're so important is because thousands of people's lives are at stake. Whenever we take a drug into the clinic, even before the clinic, like I said, when you're in the early drug discovery stages, you want to make sure you're dealing with pure substances and that the pure substances are doing what they're supposed to do before you put them into people, and even before you put them into animals. Because in vivo trials are expensive, and obviously nothing's more expensive than a human life. So you want to make sure when you get to that point, throughout the entire process, everything is documented well and everything is what it is supposed to be. So you want to make sure your structures are right. You want to make sure that multiple people go over and check your lab notebooks to make sure the methodologies are right. And you can just see how many people are involved in clinical trials. Phase 1, usually for safety. We don't even look at efficacy in phase 1. Usually 50 to 100 healthy people. We just want to see how high can the dose go until you start throwing up or have a headache. Phase 2, we look at efficacy. We start to target patients with the disease of interest. Is it safe, but also does it have efficacy? Phase 3 and 4, thousands of people are involved for safety and for efficacy. So there's a lot of people involved, and we want to make sure that with this course, you'll be better trained on what governing bodies are involved and how to maintain a good SOP, where to start, and also what types of analytical techniques you'll be using in pharmaceuticals that'll be able to help you quantify and qualify different drug substances. So with that, I look forward to meeting everybody in the lecture, and yeah, use this video to study, and there'll be more to come. So please comment, subscribe if you have any questions, and of course, like the video. So thank you, and have a good one.